Hello grade 10. Today we started with section 6 and chapter 8, gene expression and regulation. We learned before that our cells tend to make proteins to supply our needs. We need proteins for many purposes and for many functions in our bodies, like chemical reactions for building, chemical reactions for breaking down. So proteins are needed, and we said also that these proteins, to be synthesized, there are certain genes on the DNA should be expressed, and that means expression means to switch on. Okay, how this happens? Through the DNA transcription and translation. We learned that translation happens when a certain gene will be transcribed into mRNA, and this mRNA will take the message and it will be translated in the cytoplasm to make the certain protein. Now these processes should be regulated or controlled. So gene expression, which means switching on the gene, should be regulated among the needs of your body. If your body needs certain protein, the gene will be expressed. That means it will switch on. And if you don't need this gene anymore, it will switch off. Okay? So, this gene expression should be regulated. As we learned before, we have two categories of cells. We have prokaryotic cells and we have eukaryotic cells. Now, the regulation of gene expression is different among these two types of cells. Now, in the prokaryotic cells, the DNA is fewer than the DNA in eukaryotic cells, and the genes are organized in different way. Today, we will learn how they are different. Now, first, before we start, I would like to mention that the gene includes more than just a protein coding sequence. That means if you look here at this one, just 1 minute please. If you look here at this one, this is the sequence, right? On a strand of DNA. This sequence shows different nucleotides, right? C, G, U, A, okay? These are sequences. هدول sequences on the genes. Not all these genes are coding for proteins. In في عندي departmental genes, they have different functions. Not all of them are certain only for protein coding. No, there are some parts for controlling this process. في عندي nucleotides sequences that play part in controlling this expression of the gene. Okay, the start of a transcription is largely controlled by these sequences. From these parts or segments on the genes are called promoters and operators. So, what is a promoter? Promoter is a part of the DNA segment. DNA segment has a part which allows a gene to be transcribed. Control the transcription of the gene. And the operator is part of DNA that turns the gene on or off. So, next to the gene, في عندي segments of DNA are specialized for controlling the process of gene expression. These are promoters and operators. The promoters in the DNA allows gene to be transcribed. Now, this promoter will let the gene to be transcribed, and the operator will switch on the gene, okay, switch on the gene so the RNA polymerase can bind and start transcription of DNA to mRNA with its message. Okay, now if we look here at this structure of the DNA in a prokaryote, we will talk first on prokaryote cells today. Prokaryote cells, they have DNA, but their DNA is fewer than our DNA. They don't have the same number. And their genes are organized in different way. They are organized in such a way. Can you look here please closer? Now, in the prokaryotic cells, this is one strand of DNA. Here are the genes that coding for proteins. And here are segments of DNA that control this, the expression of these genes. Okay, so you can find in the prokaryotic, like bacteria, we have 3 genes. These genes are expressed for enzymes that digest lactose. هدول الجينز the 3 they are expressed for enzymes that digest lactose. يعني if the bacteria has lactose and it needs to break it down, this bacteria must synthesize enzymes which are proteins that digest the lactose. So they need to express these genes that are specialized for these enzymes. Now, these 3 genes will become as a cluster, like a group together, and they will be controlled by 1 promoter and 1 operator. So, in expression of genes in prokaryotic cells, controlling can happen for multiple genes. Multiple genes can be controlled by 1 promoter or 1 operator. While in the prokaryotic, no, each gene has certain promoter, certain controller. So this organization is called operon. Okay. Now, operon only found in the prokaryotic cells, and operon consists of promoter, operator, and genes, structural genes that located or found as a cluster, as a group. All right? Okay, let's go back to the previous page. Just 1 minute. So, operons are most common in prokaryotes, and they include promoter, operator, and 1 or more structural genes that code for all the proteins needed to do a job. They most common in prokaryotes, and one example of these operon are lac operon, which it's an abbreviation for lactase operon, اللي هو enzyme lactase. This one will break down the lactose if it present in the bacteria. So lac operon is one of the first example of gene regulation, and it has 3 genes that code for the enzyme that break down lactose. Alright, tomorrow we'll continue, guys. See you and take care.